So while Tatiana is getting mic'd, I just wanted to again say thank you for being here. We're going to start this session with kind of a little bit of a roadmap from Tatiana, so she can sort of walk you through kind of our vision for the session, and we'll be introducing speakers as they come up to present. So with no further ado, I'd like to introduce Tatiana Rynearson from the University of Rhode Island's Graduate School of Oceanography.
All right, good morning. So what we're going to do is we're going to mix it up a little bit. I'm going to spend the first 5 minutes giving you a little bit of an intro to this session, as well as a roadmap. And I have 3 main goals for this session. The first one is to give you a sense of some of the processes, some of the ingredients that are involved in contemporary evolution. So you can get a few of those terms under your belt. My second goal for this 5-minute introduction is really to give you some important tools so you can avoid that awkward interaction you might have at your next cocktail party when you're cornered by an evolutionary biologist. You'll know some of those terms, and you'll be able to say the right thing, perhaps get them thinking, and slip. So that's my second goal. And the third goal, and I hope this continues through the session, is to highlight that often we think about evolution as something that happens very gradually. So this takeoff on that fairy poster, very gradual change. In fact, that change can actually be quite rapid, especially in some of the microbial populations that we know drive biogeochemical cycling. Okay, and I know it's first thing in the morning, and there may be those of you in this session who showed up and were like, oh shoot, I could have slept in. They're talking about evolution, right? I don't know anything about it. And so I want to start with what might be a happy place for most of you, right? So here we have carbon cycling in the biological pump. Very familiar figure, a classic figure of what might be going on, just to highlight the biological pump. One thing we generally think of in oceanography is often we're thinking about the things that are influencing that biological pump, both biological and environmental pressures like temperature, nutrients, predation. And we're often thinking about how that changes the ecology and how that ecology then has ripple effects on biogeochemical cycling. What we're going to talk about in this session is evolution, and the idea that we might want to explore how evolution influences the ecology of these systems and how that then may influence the carbon that's transported to depth or respired. I have an arrow up here to predation to indicate that when we think about evolution, we should also be thinking about ecology and the tight and intimate link between them. So you can think about eco-evolutionary dynamics. And of course, things that we know are going to be changing in response to climate change are also going to provide new kinds of selection pressures on these organisms, which is going to drive evolutionary change, which then may have influence on ecology and ultimately biogeochemical cycling. Okay, let's start with some ingredients to evolutionary processes. So here we have a phytoplankton cell. We're thinking about evolution, contemporary evolution. We can think about groups of cells or populations. So you can picture this as a whole population of cells. The fundamental change that happens to these organisms is mutation. Once mutations are present in a population, they can be acted on in several different ways. So the first is recombination. What that does is it mixes up the genetic mutations on different genetic backgrounds. Then we have migration. Migration introduces new mutations to populations, and it also mixes up those genetic backgrounds. Okay, and then we have natural selection. Natural selection, of course, will influence the fitness of different organisms and thus their survivability. So we're really interested in fitness of these organisms in response to things, to pressures, selection pressures. And then the final ingredient, see how easy evolution is? Just 5 ingredients. The final ingredient is genetic drift. And so genetic drift is a process of random change in the frequency of those mutations over time in a population. And it's most important in small populations. So we're really interested then in population size in terms of understanding the influence of these kinds of random processes in the patterns that we see. Okay, so that's pretty much it. You can all like walk away and have that cocktail party conversation with an evolutionary biologist at this point. Hopefully you'll also be able to better understand the talks that are coming up this morning. We have two kinds of sets of talks. The first set is looking at experimental evolution in the lab. These are run under very controlled conditions where we can control the kinds of selections that take place. They allow us to measure the types and rates of mutation. And importantly, they allow us to directly measure fitness. So Lisa Schaum will kick things off by looking at a single species and evaluating what's happening in that species in response to selection. Jeffrey Morris is going to come in and look at species interaction and the evolution of interactions, also in the lab. And then the second half of this session will be focused on the spatiotemporal patterns of contemporary evolution, looking at the standing stock of diversity and looking at the results of that selection, and in two very different ways. The first is my talk, where I'll be talking about single species. In that case, I look at very specific locations in the global ocean and take living cells out of there and have them report back as to their evolutionary histories and potentials. And then the second talk will be Victoria Coles, and she's going to look at emergent species in silico. So she's going to be using a modeling approach and a set of equations to see if we can understand some of these processes through a modeling approach. So I hope you enjoy the session, and so it's my great pleasure to now introduce our first speaker of the session, Elisa Schaum.